Today's subpoint under the unit number 4 plant breeding, it is the pure line selection. In a pure line selection, we'll see the introduction, definition, procedure, merits, demerits, and achievements. The pure line selection, the term was coined by the Danish botanist W.L. Johansen in 1903 for the progeny obtained by single homologous plant is more and more homozygous nature for the selected characters can be obtained by a self pollination for a few generations till the character shows consistency at least for 3 generations. A variety developed by a pure line selection shows more uniformity in the characters than a variety obtained through mass selection. The pure line selection is also called as individual plant selection. The individual plant is the best. The progeny selection, that is the selection in further generation, is done on the basis of performance of the progeny of individuals. Single line selection, in a single individual plant selected and their progeny is maintained separately. Head to row selection, in some plants instead of plant only the head is selected and the grains in a selected heads are sown in a separate rows. The pure line selection is carried out for the purification of characters on the line basis, and ultimately the development of variety is achieved with a pure line characters. The procedure: in the first year, few plants having a desired characters are selected from the mixed population in the field. The seeds from each selected plant are collected separately and are sown separately in a different rows. During second year, by selecting progeny individuals and by repeating the procedure from third year to 8 years, superior individuals with desirable characters are selected. During next 2 years, the seeds of these individuals are multiplied for commercial distribution. In this process, the replicated yield tests are carried out at a different location, are repeating them for 2 to 3 successive generations. Most suitable varieties are identified. The breeders, on the basis of comparative performance, select 1 or 2 strains of the variety and gives a suitable name to them. These strains are multiplied and then distribution for commercial cultivation. It is generally adapted for the improvement in self-pollinated plants. The variety developed by this method is with a uniform genetical characters and long-lasting nature. It is also useful for hybridization technique. In case of cross-pollinated plants, the variety mixed by the farmers can be separated and purified with a pure line selection. Steps in a pure line selection method. In the first year, 200 to 3000 plants are selected from superior phenotype. In a second year, the individual plant progenies are grown, undesirable progenies are rejected. In a third year, the preliminary yield trials are carried out. The selected progenies are planted in a preliminary yield trials and inferior ones are rejected. The 4th to 6th year, the multi-location yield trials with suitable checks are conducted. Disease resistance and quality tests are done. And in the 7th year, the progeny superior to check the variety is released as a new variety. Seed multiplication begins for distribution. Merits of pure line selection method: to improve the local varieties of self-pollinated plants. It is a simple and easy as compare the plants to be hybridization. It does not require emasculation and crossing. The variety obtained by this method shows uniformity in characters and good performance. The method can be used for both self and cross-pollinated crops. Then demerits: no new character can be introduced by using this method. Once the pure line is established and isolated, further generation is ineffective. Albino variety, low yield, and dwarf forms are some of the undesirable characters found in such varieties. Such varieties are susceptible to adverse conditions like a drought and pest attack. The varieties are homozygous and unable to adapt the variations in the environmental conditions. There are some achievements by using this pure line selection method, that is the in a wheat NP4, NP6, K13 and K54, while in a rice ADT1, ADT3, while in groundnut TMV3 and TMV4, while in sorghum CO1, CO4 and CO